Okay. Well, good morning. I'm Dennis Ray, and I'll lead the feedstock development and production session this morning, along with Colleen McMahon, who is my co-lead in this. I will chair this session, and Colleen will take notes and see about themes and things and questions that we want to cover in our session on Wednesday morning. If you have questions, of course, go to the bottom of the Zoom page to the chat, and Torin will keep track of them. If we have time, if you just hit the raise your hand function under participation, and at the end of something, if we have time and I see that, I could call you directly, but probably the best way is to put them in the chat, because then they stay as a record. As you can see, we have quite a few PIs in this area, and I have to say it's been my pleasure to work with this group over the last 3 years. As I look back to where we started, I can definitely say that we have made significant progress. As you can see on the next slide, our group covers both feedstock development and sustainable feedstock production. Under feedstock, feedstock is the foundation of sustainable production. I'm not saying that it is the most important part, but it is the foundation upon which the other work is based. Under feedstock development, we are doing genetics and breeding of feedstock, phenotypic characterization of the feedstock, work to reduce and eliminate flowering in Wayuli. Under sustainable feedstock production, we have projects on irrigation management, herbicide tolerance, expanding the growing range of the feedstock, determining optimum planting densities, and soil health. In the next number of slides, I'm going to touch upon highlights from these areas. This is going to be fast and not comprehensive. These slides will be available to you, and if you have questions, please ask me or Colleen or the appropriate PI, or if you're not sure to ask, put it in chat and we'll get it to the appropriate person. I'm not going to read all of these. I'm just going to try to hit high points because there are a lot of words and it's early in the morning. As you see on this slide, we have highlights of GUAR through year 3. We've completed the first year of the regional variety trial, and the second year is in the field, is planted. The good news is we found that yields in the first year are above the economic threshold needed to bring GUAR into production. Irrigation studies in GUAR show that it is an ideal semi-arid, arid lands crop, with 4 irrigations being enough to give economic yields. Density studies in GUAR and Wayuli are quite similar. You have lower yields per plant at higher seeding levels because the plants are crowded together. You have higher yields per plant at lower seeding levels because the plants have more space. But the plants compensate for this space in various ways, and the yield per area is essentially the same. So we're finding that this is good because it gives us quite a range of how we can plant both GUAR and Wayuli. Salt tolerance work has just begun in GUAR and shown that differential expression of transporter genes and roots are important in salt tolerance. In the next slide, you'll see that these studies will continue. That's the next slide, please. Thank you. That these studies will continue in year 4. Now, for the Wayuli, we'll go to the next slide. You can see that the second year of the regional variety test has been completed, but as of yet, not all of the year 2 data are analyzed. Because Wayuli is a perennial crop, irrigation studies are also continuing. You will hear more about these studies later. But again, we do not need to over-irrigate Wayuli. We do know that rubber and resin content per plant decreases with increasing water application. Also, soil types affect irrigation management. We need different amounts of irrigation in different soil types. The results of the density study is much like in Lohr. You get the same yields per area with differing plant densities. The question becomes, since there's a cost for seed, at what seeding rates are yields economic? In the next slide, we can also look at salt tolerance in Waiuli. Plants and lines that were initially tested in the greenhouse have shown salt tolerance have been transplanted into the field in order to increase seed for future generation testing. Herbicide tolerance is extremely important, and we have come a long way thanks to Bill McCluskey. We now have some herbicide sequences that show excellent results. Again, I'm not going to go into this, but if you would like to ask questions of Bill, he is available. In the next slide, you'll see the results of our work to reduce flowering and the soil microbiome. There will be talks on each of these areas, so I will be brief. For the work to reduce or eliminate flowering, transformants have been made, and there are thousands of transformations being tested at this time. The soil microbiome work is fascinating, and the results are beginning to come in. Interestingly, they are finding that the root zone microbiome is significantly different during plant dormancy and active rubber production than it is during active vegetative growth. Again, more later. In the next slide, you will see that all of these projects will at least start in year 4 for Waiuli, but several of them, the yield test, the irrigation studies, and the density test will come to an end, although there is one more irrigation test that we've started. In the last slide, you'll see our 6 presenters for today. These are very interesting people. I found out something about each of them, and they are a diverse group with diverse interests outside of this work. For instance, a number of them like hiking, reading, doing jigsaw puzzles, ethnobotany, even one teaching himself Python. I apologize to the presenters for not including all the interesting things they do, and forgive me if I miss some details below. We'll start with Blase Evancho, who is a graduate student, but also in U of A Extension. He will be talking about Waiuli root growth and the start of some nitrogen studies. However, much of his time recently has been taken up with the birth of his son, Blase Jr. In fact, talking to Blase a little earlier, we know that he only got a couple hours sleep last night, so be gentle with him. Alonso Garcia has graduated with a master's degree in plant and environmental science from New Mexico State University and is presently working on his family farm in Guerrero, Chihuahua, Mexico. He will talk about irrigation work in Lohr. Hadiqa Maqsood earned her bachelor's degree in Pakistan in civil engineering and a master's degree in England in water and the environment. She has served as a visiting faculty and an assistant professor, worked on a USAID project, and is now working on her PhD at the U of A on a Fulbright scholarship. She is coming to us today from Germany due to travel restrictions during this time, and will talk about Lohr and Waiuli irrigation. Leah Ossanna is from Tucson, has earned her bachelor's degree in environmental science at the U of A. She is presently a research specialist here and will talk about the soil microbiome work. Mariano Resendiz did his bachelor's at UC Riverside in plant breeding. He just finished his master's degree from Cal State East Bay in computational statistics. He has worked as a plant breeder, a lecturer at the college level, and is now a research assistant at the USDA Western Regional Research Center in California. He will discuss the plant transformation work. Patrick Lohr grew up in Tucson and received his bachelor's degree in chemical engineering from the U of A. When he finishes this degree, he will continue on for a PhD, but not sure in what area, but he does really enjoy programming. He will talk about modeling growth in Waiuli. I'll have a few minutes if you have any questions, but otherwise we'll move on to Blase.
Great, thanks so much, Dennis. And also, Colleen, thanks for that suggestion. She was just saying we're running behind time, so if each of the Feedstock presenters could stick to 10 minutes.
All right, I could do better, one better than 10 minutes. I'll be under. Hello, everybody. I'm Blase Evancho. I'm going to briefly go over a couple studies that focus on the agronomic investigations to improve Waiuli production. Next. All right, so here's the graphic put forth by SBAR. I'm sure many of you are quite familiar with. On the right there, we fit in, as Dennis was just covering, into that number 2, the second stage of this pipeline, and we're focusing on plants and optimizing production of Waiuli. So we want to develop a better understanding and improve management strategies of Waiuli and this great feedstock, so that we could really expand on the economic driver of this whole feedstock and this whole breeding pipeline. So really what we're doing in our lab and a lot of our research, as Dennis mentioned, focusing on improving weed management strategies to allow us to produce it better, a better increase or a better understanding of irrigation strategies so that we could be more refined in our use of water. And overall, what I'm focusing on is just an increased understanding of our agronomics and what it takes this plant to grow, so that we could eventually have the most efficient and highest economic potential for this bioeconomy with Waiuli. Next. So the first experiment that I'm going to cover briefly here is this rooting structure comparison that I'm doing. So I'm establishing Waiuli in 2 different methods. I'm growing them in small plugs and then transplanting them, and I'm also direct seeding them into these large pots. So they are 120 centimeters tall by 25 centimeters round. They are filled with a loamy sand from the field. They come in at about 250 pounds when they're freshly watered, so they're quite difficult to move around the greenhouse when I'm growing them. And what we do is we grow these plants. I establish them in the pots, and then after about 30 days, they start to grow more rapidly. So when they start to grow more rapidly, then we set out basically a 6-month harvesting schedule occurring about every 30 days in that 6 months, where we destructively harvest these roots. And if you look on the picture on the right, you can see how we go about doing that. So first thing we do is we wheel these big suckers out into a field, and then we cut the pots in half. Then we slowly use water and our hands to massage away all the soil. So that's what you see in that second picture on your right. You can see the bare rooting structures of that whole big pot. And then at the bottom, you can see the dried roots and dried above ground biomass that was produced by that plant. So you see there's 6 different sets of piles there. Each one of those piles represents 20 centimeter section going down through the pot. So we do this so that we can compare root growth down through that soil profile between the direct seeded plants and the transplanted plants. And what we're doing here is collecting above ground biomass, collecting rubber, resin, and then all the different above or below ground biomasses. And we're also looking at how the root structures differ from a morphological standpoint. Next. So here's, I'm going to give you just a quick synopsis of what these plants go through over a season. This is the very beginning, right? This is 30 days after they started to rapidly grow. You look at these two, you could see that there's a little bit of difference in the above ground biomass of that top plant, which is the transplant. And then you can start to see that the rooting structures are starting to look a little different. There's 2 main roots on the top one, but the bottom one pretty much just has 1 root. We found that the root mass or the above ground biomass can be a little bit different statistically at the beginning, but after a couple months they catch up and then pretty much grow the same rate for the duration of the experiment. Next slide. So then here it is. This was our last harvest from our first experiment. You can see this is 226, or about 6 months after planting. And you could see the dramatic morphological difference in these rooting structures. For example, that top plant has about 6 or 7 different coarser roots, and that bottom one just has one main taproot that goes down through the whole profile. You could also kind of pull out that some of those thinner main roots from that top plant grow more to the side of the pot instead of straight down, whereas that bottom plant really shoots straight down through the profile, and you see a couple of lateral roots eventually going out to the side, but for the most part, it's penetrating deep into the soil. Okay, next slide. So what we really pulled out of our analysis of the data after these 6 months was the above ground biomass, the rubber production, the resin production. They all pretty much maintain the same development and same quantities produced. There's not a statistical difference except for a little blip in a couple of the data points, but in general, it's pretty straightforward. What we do see is a very different morphological difference, as you can see on the right. That top transplanted plant just has a completely different structure than the bottom one. So really what we could look at in the future on this is trying to understand how these plants use water differently. Do they go for deeper depths? Do the more fibrous transplants spread out closer to the surface? And how does that really affect water use and uptake over the lifespan of this plant? Next slide, please. So then here's our nutrient requirement study. So I'm looking at, eventually we're gonna look at nitrogen and phosphorus, because it's really hard to get a good understanding in the field of what's going on with these plants, because we're always having releases of nutrients from the soil. We're adding nutrients through our irrigation water. We're adding nutrients through rain. There's crop residues from other crops. So there's all sorts of different things in the field that are manipulating the Waiuli. And since it has a very low nutrient requirement, being a desert plant, it's hard to get a good understanding of what an optimum fertilization level in the field is. And then we're going to try some different rates and see if that affects not only above ground biomass, but some of the other co-products associated with the plant. And really what my goal is here is to develop a nutrient response curve. So we're growing these hydroponically pretty much. And then I want to see from the lowest levels, what kind of above ground biomass is produced and what do those nutrient levels look like in those plants. And then try to develop a high end of what the maximum rate of biomass production at the lowest amount of nitrogen or nutrient availability is. So if you look at that picture on the right, you could see 2 larger plants in those first 2 pots on the left. You could also see a really small stunted plant on your right with a blue stake behind it. That blue stake represents the lowest nitrogen treatment in our study that's currently in the greenhouse, but has kind of been not well taken care of because of the COVID. We kind of had to let it go and put it on life support for a long time. And you can see that alleyway down the middle. Those are our 6 different tubes delivering 6 different nitrogen solutions, which I'll cover on the next slide. So we have originally set this trial up in number 5 pots of perlite, which is an inert soil media. All the nutrients basically flow through it. It's just something for those roots to grab onto. And we deliver hydroponic solutions of 6 different nitrogen levels. Originally, the idea was to go from 25 ppm, which is about as low as we can go, up to 200 parts per million, which is a common amount of nitrogen delivered to a commercial crop of wheat when we do experiments, and then kind of give it 6 different rates throughout there. The only one we really saw in that first trial in the greenhouse, we only saw the nitrogen difference in that lowest pot level. The rest kind of look the same, but we haven't analyzed any of these numbers yet. So we'll have a better understanding of exactly what those numbers look like here, probably in the next couple weeks. And then you can see how they're randomized throughout that greenhouse on the plot plan on your right. There are 12 reps, which 6 different plants in each. Those colors correspond to different nitrogen treatments. So we just want to—
Blase, this is Colleen. You're down to 1 minute, okay?
Okay.
Thank
So we
you.
really want to just develop a good understanding of what's in those leaves and how they're corresponding to growth. And hopefully this will let us elucidate that a little bit better. All right, and I think that's it for me. So thank you everybody who's helped me. Thank you, young Dennis in that top left picture. And thanks to the countless people who've helped me muck around in the mud in the field and wash these roots.
Great, good to see young Dennis. And there are some questions for you in the chat, and I'll email them to you so you can respond to Bob and Julie when there's more time.
Okay.
Okay. Thank you, Blase. Alonso. Alonso, are you're muted right now? Unmute yourself. There we
Oh,
go.
right there, okay. So good morning, everyone. Thank you, Dr. Ray, for introducing us. As he was mentioned, I work at, I got my master's at New Mexico State University along with Dr. Grover, studying the GWAR. So I'm really excited to talk a little bit about what we did during these 3 years. Can you go to the next one, please? So why WAR? As part of the SBAR project, is that we establish different crops in which they can tolerate the heat or have low water requirements, because in our region we don't have that much water. And I like this picture on the right because it shows that, for example, on the yellow to orange colors is between 5 to 10 inches of annual precipitation from 1961 to 1990, and on 2016 and 17 we have 6.24 inches of water. So water is reported to require a low amount of water, and it could be a good crop rotation from chili, onions, and it's a low cost crop. Can you go to the next one, please? So one of our objectives was to evaluate the performance of WAR under 5 irrigation regimes. We have drip irrigation system. So we look into these 5 treatments we started after the first month, because as we know, there's several studies that they discuss about the early stages that are more critical, but we wanted to show these studies from the flowering and after flowering or in pod formation stages. For the first irrigation regime, it means to irrigate the whole season. The second irrigation regime, we keep irrigation at 75% pod formation. The third one was to skipping at 50% and 75% pod formation. And then for the irrigation 4 and 5, we terminated the irrigation. All these treatments we started after the first month or after the establishment of the crop. And the difference between irrigation 4 and 5 is that for the irrigation 5, we introduced this biogenic silica. I'm originally was born and raised in Mexico, and some of the agricultural practices that we've been trying to improve new techniques, and we've been using the biogenic silica as a plant growth promoter. Biogenic silica is the same as diatomaceous earth, and as far as I know, there's more research for the biogenic silica to control insects. But there are some studies because the biogenic silica is a complex form of glass, and it helps with the absorption of light. And in the case of the plants, it helps with the stomatal opening and closure, and the tracheids for the transport of the nutrients. It's made of silica. Of course, there are several plants that they absorb the silica, and the Equisetum family or the horsetails are the only family that they actually it's necessary for their growth. And then these are some pictures that because we have those beds, and then we have the drip tape, and then we have some sensors installed in the field, and this is there are some pictures you can see on the screen for how the biogenic silica looks, and then the GWAR at flowering when we started our treatments. Can you go to the next one, please? So we measure in different sets of things during the 2016 up to 2018. For example, the canopy width, branches per plant, above ground dry biomass, leaf area, dry leaf weight, and different pod parameters. For example, pod width, pod thickness, cluster per plant, 1,000 seed weight, harvest index. But we wanted to focus, since we have a short time, we wanted to focus on some key results as suggested by Dr. Dennis Reid, and thank you for helping us in our previous meeting with that. So I'm gonna explain a little bit with some work that we did with the light core and the yield. So in this table, it's pretty simple. We have all the different treatments, and as we can see, as we skip more irrigation, for example, at 50% and 75% pod formation, and when we terminate irrigation at flowering, that was significantly lower compared to the irrigation throughout the entire season. But for example, what we can see interesting in this table is that for the photosynthetic rate, as well as the stomatal conductance and transpiration rate, the irrigation treatment number 5, when we terminate irrigation at flowering plus biogenic silica, with the addition of the biogenic silica, we can see that it's still among the between the irrigation throughout the entire season and the terminate at flowering. So it's still higher the values from compare those two treatments. Can you go to the next one, please? And on this the last table, on the x-axis, you can see the years, the 16, 17, and 18, and on the y-axis, it means the seed yield for kilograms per hectare. And again, I think we found two things that they were very interesting. The first one is that the guar as a crop was able to survive for the whole season by only irrigating the first month. And repeating again, we wanted to skip the irrigation after the first month because that's when the rains begin in New Mexico, in Las Cruces. And then as you can see, it was only significantly different with the biogenic silica treatment. That's the blue bar, and in the irrigation 5. And then on the 17 and 18, it was not significantly different compared to the the terminated irrigation at flowering and terminated irrigation at flowering plus biogenic silica, but it was still higher the seed from those 3 years. So I think that was something very interesting. So that's why we think that guar is an potential alternative crop in southwest New Mexico, and it meets the SBAR project. So I would like to thank the SBAR and IFA for the support, because for the funding for all those activities and the meetings that we have together. Of course, Dr. Robert, my advisor, because he's been always helping with me, the patients, and then we work together along to achieve our objectives. And of course, the New Mexico State University staff and the colleagues for being all together on this project. And I think that's pretty much. I don't know if there's any questions.
Are there any questions?
Nicely done. Oh, sorry. We have about 2 minutes if you want to take a few minutes for questions. Nicely done, Alonso. I'll ask one question, and that is, what you mentioned a couple of crops that biogenic silica is used for. Could you expand on that, please?
Yes, what I mentioned is that only for the Echisetaceae family is essential as an element for plant growth, but it's not necessarily an element that it's how is it called? That it's required for a plant growth. But it's more like a plant growth promoter. But it helps with the physiological things on the crop. That's how we found. Another thing we found interesting is with the SPAT, the chlorophyll content, it increased with the use of biogenic silica. So it helps with the absorption of light. But it's only essential for the Echisetum family. The other plant, it has not been prove that it's actually necessary for the plant growth. It's more like a plant growth promoter.
Thank you.
Okay. Hadiqa, are you with us?
Hello, yes, I am.
Go ahead. Thank you.
Thank you, Dr. Ray, for introducing. Hi, everyone. I am presenting on behalf of my lab, where we have worked on the soil moisture and irrigation models and sensors and remote sensing techniques for Wylie and Guar. So I am going to introduce the whole Can I have the next slide, please? All right, so we are meeting the SBAR goals of Wylie and Guar, so increasing the acreage, and then the tools will be helping to develop and distribute the production in it. So can I have the next slide? So just to do an overview of the field experiments, on the left side, you see Wylie, which is planted in Maricopa. We have 1 flood irrigation treatment there, and then 5 drip irrigation treatments, starting from 50% of drip to 150%. And then on the right side, you see Guar field, which was planted in Clovis, New Mexico. And that was on center pivot sprinkler irrigation system, hence the 2 wedges. And we had 2 initial conditions. So that was, one was pre-irrigation and the other was no pre-irrigation, which means water was not given to the plant before sowing. Then we have 4 in-season treatments starting from full irrigation. Then we stressed the crop at 2 different stages: early stage, that was vegetative stage, and then stressing at reproductive stage. And the 4th one was rain-fed, where no irrigation was given. So in my next slide, I am giving you a mind map of what we were doing. The blue portion is all the water modeling part, where we use the winds model that was developed by Dr. Waller, my supervisor. And that gives us soil moisture analysis and crop water requirement. The green portion is the remote sensing component, where we are using X4D model to do the image processing and generating some 3D models and trying to estimate crop heights. The gray part, which is the crop behavior, is we are trying to integrate both these components and get some results on it. So in my next slide, I'm showing my the first progress, which is on the sensor. So that's Matt with the sensor. This is a sensor package developed by Matt and Danielle. There is a TDR, which is below the soil. And then we also have a camera, which is above the ground. And together they are providing some some in-field measurements for the crop as well as the soil. So far, this has been successfully installed in the at home due to COVID. And there is a progress that the enclosure was in 2 different parts, and now it's 1 single enclosure. And then we are in the stage of developing 6 such packages that will be deployed in eloid irrigation experiment. So coming to the next slide, can I have one more click? All right, so here I am mentioning about the results of the winds model. The winds model has been successfully calibrated for both the crops, Wylie as well as Guar. One unique part of winds model is that it does layer-based analysis. So we have the water content for each layer. Generally, we have like 20 centimeters interval for each soil depth. And on the right side, you see a graph, which is one example from a Wylie experiment. And it's showing that the blue dots, which represent the neutron probe readings from the field, and the line, the gray line, is the daily simulation from the winds model. So you can see how they're in close proximity and they're following the same pattern, which means the model is responding pretty well to the field results. In another slide, the next one, I am showing one more example. It is from the GWAR experiment. So these are also results from the winds model. And you can see I'm showing the solid line, which is pre-irrigation condition, and the dotted line is the non-pre-irrigation condition. This is for the root zone of GWAR in 2018. And it's telling us that initially the water was quite different, but overall within the season, it kind of followed similarly. And we have these for all the different treatments and all the layers for both crops. Now coming to this slide, I'm talking about water application with the yield. This is an example from Waiuli experiment. Okay, so if you see the solid black line is drip from Maricopa, and the dotted black line is drip irrigation at Eloy. Now, if you just look at the highest point of both the lines, we have the highest of Maricopa, the solid black line, at 100% of irrigation. Yield coming from Eloy is at 75%, which means that there is a difference in soil type. And for clay soil, which is at Eloy, we are getting good enough yield within 75% of the water given. However, Maricopa compared to Eloy. In the next slide, I have similar results for GWAR. Now, just to tell you, the leftmost dot or leftmost point is the rain-fed treatment where no irrigation was given, and the extreme right is the full irrigation treatment. So, and the black dot, the black line is 2018. The dotted lines are from 2019 experiment. So the results are showing that the 2019 experiment did not work very well, and we are trying to do some reasoning to see what were the reasons. And somehow there were more rainfall events. There was a little bit more of irrigation as well in 2019. And to see the seed yield, we have rain-fed and stressing the crop at reproductive stage, giving us the highest yield if we do pre-irrigation. And this is pretty interesting. It just shows that GWAR responds well to stress. Now, in the next slide, I have remote sensing results. So the left side of this slide, you see Waiuli experiment, and the right side is again GWAR. And this is showing a vegetation index, which is telling us the live green vegetation. So the greener portion is healthier and live green vegetation compared to the red portion, which is lighter in vegetation and not as dense. In the next slide, I will be further telling you about how we have used Pix4D. So the left side is showing a zonation map, which could be very useful for farmers where you can evaluate your field and try to figure out which zone is prone and which zone is healthier. And on the right side, this is a 3D which I developed to estimate the crop height. So because some of the crop height data was not calculated, was not measured in the field. So using remote sensing is a very good way to still extract the data from the field. In the next slide, I am now, just to summarize the whole process and the work that we have been doing. The winds model, which has now been calibrated, is being validated on 2019 data. And then we are using the infrequent to work on the calibration and then use the calibration estimate the daily moisture content. And based on that daily moisture content, we will work on the effect of stress on the crop for which one works and which stress working best on the crop. As we saw for guar, stressing at reproductive stage worked really well. Then for crop coefficient part, we have developed Waiuli and guar crop coefficient, not updated. The third part, this meets the SBAR objective of developing tools for growers, and remote sensing has worked really well in terms of telling us and quantifying us the water stress for the crop and showing us more extract from. Good.
Hadiqa, you have 1 minute.
All right. All right, so just this is coming to my last slide here. I'm talking about the future research. So our group is working, which will incorporate all these components in the mind map that I showed you. And then we are also going to incorporate the data for 2020 and use the remote sensing as well for that. In the next slide, I want to thank everyone for listening. And if you have questions, please let me know.
All right. All right. Thank you, Hadiqa. Thank you very much. And good evening to you. We appreciate you coming on this evening.
Yeah, it's almost 7:30 here.
Yeah.
PM.
Very good. Okay. I don't see any questions, so let's move on. Leah?
Hey everyone, thanks for being here today. My name is Leah Ossanna and I'm presenting on behalf of the Mayor Lab today. And also I'd like to recognize that our work is done in collaboration with all of these other nice folks on the screen. Next, please. First of all, the term soil microbiome refers to the bacteria, archaea, and fungi that exist in the soil, including the soil that surrounds the plant's root zone, as you can see pictured on the right. If we can understand how soil microbes interact with a plant, we can alter the soil conditions and therefore microbial community to improve the Waiuli crop production. Plant microbe interactions for other production crops are already commonly employed, such as utilizing rhizobia to enhance alfalfa growth. We want to do the same for Waiuli. Next slide, please. The Mayor Lab is currently working on 3 Waiuli microbiome studies. I want to stress that this is the first research of its kind. So despite that Waiuli has been studied for decades, examining the soil microbiome is actually a novel approach. So hence the goal of these studies is to provide background knowledge to inform us on how to design more specific studies in the future. Our 3 studies focus on winter dormancy rubber production, fungal pathogens, and the Waiuli associated core microbiome. Next slide, please. This first study is about winter dormancy and rubber production. So for this study, we collected paired plant and soil samples from the Maricopa Ag Center fields at 3 different time points to represent the transition into, during, and out of winter dormancy, which is when rubber is produced. Previous studies have demonstrated that Waiuli rubber transferase activity spikes once night temperatures drop below a certain level, as you can see in the top right-hand figure. The sharp increase in rubber transferase activity, which is represented by the blue bars, coincides with a decrease in the red line, which is minimum night temperature. And so you can see the highest date is, the highest bar is Julian date 352, which is in mid-December. And the graph continues through 436, which would be mid-March. The increase of rubber transferase activity indicates that rubber biosynthesis is beginning, and with this data, our 3 sampling dates correspond to these different phases of plant growth. The bottom 2 graphs are of data collected from this study. So the graph on the bottom right shows that we successfully captured the increase in rubber transferase activity by sampling in November. And the graph on the left is of rubber content, which is percent rubber by weight. And in this study, you'll see that we focus on rubber content or percent rubber as our rubber metric because For us, that's the most relevant to economics of Waiuli production. The rubber content graph shows that rubber accumulates over the winter dormancy growth phase. And from both of these plots, so the grouped stacked bars are of 3 different plots in the field. And so you can see that there's actually variation in what are identical plants supposedly experiencing the same conditions, because this all came from the same irrigation treatment. Next slide, please. But actually, from what we see is that the conditions are not the same. And when we look at the soil microbiome, we see that the plant is actually experiencing different conditions, both temporally and spatially. So first, from the top right-hand graph, this shows that there's a significant increase in DNA biomass from November to February. And DNA biomass includes the soil microbes we talked about and also plant residues. We also performed amplicon sequencing on the extracted soil DNA to characterize the microbial community. The bottom 2 graphs are ordination plots of beta diversity, and each point on the plot represents a sample. And you can see from how they cluster that it clusters according to sampling time, which is divided up by the different colors and also by the ellipses. So both of these plots show that the plant is experiencing a shifting soil microbiome throughout the winter dormancy period, hence the different colored ellipses and clusters are not completely overlapping, and also that spatially across the field, plants are experiencing different conditions. You can see from the bottom right-hand graph that the February, the orange, is a larger ellipse, and so the microbial community is less similar to each other. And based on this, we think these changes are likely to affect plant growth, these differences in the microbial community, and possibly these differences could also affect rubber biosynthesis. Next slide, please. So we looked also at correlations between soil and plant metrics. The red dots are negative correlations and the blue are positive. And if you look at the bottom row, indicated by the green arrow, that you can see that there is a positive correlation between soil DNA biomass and percent rubber. Next slide, please. So this slide is a heat map of more correlations. It's kind of complicated, so I'll walk us through this. First, if you look along the bottom axis, you see that these are the same soil and plant metrics that were shown on the graph just previously. And on the y-axis is a list of bacteria families. And the actual names of these families isn't important, so don't worry about that. But know that these families specifically were picked from hundreds of families identified in the soil samples, because these are ones that drive statistical differences between each growth stage as determined by LDA effect size analysis. The heat map itself is correlations between the rubber metrics and the relative abundance of these bacterial families. The red arrow at the top of the heat map points to the column of correlations with percent rubber, and all the families in the red box show significant positive correlations with percent rubber, as demonstrated by the black stars there. And if you look to the left at the white stars, we can see that also for most of these families, they're also negatively correlated with shrub mass and NDVI, which stands for Normalized Difference Vegetation Index, and was discussed in the previous presentation. Next slide, please. The bottom half of the graph shows that the opposite pattern is also true. So families that correlate negatively with percent rubber, that are less abundant when rubber content is higher, those shown by the white stars, those families correlate positively with shrub mass, and DVI. Next slide, please. So overall, from this study, we found that the Waiuli plants are experiencing a changing soil microbiome during their growth phases, even within the same irrigation treatment under what is, in theory, identical conditions. And there's a significant increase in soil DNA biomass after the onset of winter dormancy, which is not expected because as the plant is dormant during winter dormancy and growth slows, it is likely producing less root exudates that would stimulate microbial activity. So this is something that we didn't expect to find. We also identified plant and soil metrics that correlate with percent rubber. And lastly, we found that there are 10 distinguishing root zone bacteria families that are positively correlated with rubber production and 5 negatively correlated. And these families could be targeted in future greenhouse studies to explore mechanisms of how soil microbes affect Waiuli biosynthesis. Next slide, please. Our second study is led by graduate student Kyle Brown. He has identified several putative Waiuli fungal pathogens based on the literature, and will look at their relative abundance under different soil conditions by sampling the irrigation trials at both Mac and Eloy Bridgestone fields. Kyle will analyze correlations between pathogen relative abundance and the soil factors listed below, and he'll produce heat maps similar to the one I just showed. And understanding the soil conditions in which specific Waiuli pathogens are more abundant will be of great value for guiding future studies, such as targeted inoculations of specific pathogens. Next slide, please.
Leah, you have 1 minute.
Okay, thanks. And finally, our last study is led by postdoc Yongjin Chen, and he's working on characterizing the Waiuli-associated core microbiome. The core microbiome is a group of soil microbes consistently associated with the plant in high abundance, as you can see from the figure on the left. So this graph shows that the core microbiome members are taxa that occur frequently over many locations and that are also highly abundant. Next slide, please. In this study, we sampled frequently from the Eloy Bridgestone fields over the winter dormancy period. And the limitation from my study was the small sample size and the fact we only had one time point during winter dormancy. But you can see Yongjin's sampling plan is more robust, so we'll be able to gain a much better understanding of the potential microbial interactions using complex statistical analysis. Next slide, please. So in summary, at the Mare Lab, we're using cutting-edge techniques of DNA amplicon sequencing to characterize the soil microbiome, which we can manipulate to improve Waiuli production. Thank you so much for your attention, and I can take any questions if there's time.
I don't see any questions. Colleen, do you see any? Or
I do not. We can add them to the chat later.
okay, thank you very much. Part of the interesting as far as this study was, this came out of interaction between people talking about what they were doing. And so it's one of the exciting things that we get, having a very diverse group of researchers who talk to each other. So that's very good. Mariano, whenever you're ready.
Today, I'll be discussing the current work we have in progress concerning the downregulation of the floral identity genes in our Waiuli. Next slide, please. Waiuli is a xerophytic perennial woody shrub native to the southwestern U.S. and northern Mexico that serves as an alternative source of hypoallergenic latex and natural rubber. For Waiuli to be amenable for commercial production, the rubber quality and quantity needs to increase, as well as the genetic diversity. Thus, we are interested in the genetic engineering of Waiuli to meet this goal through agrobacteria-mediated leaf strip transformation and floral dip transformation. These transformations, if successful, have the potential to downregulate flowering to improve yield. Next slide, please. We hypothesized that a reduction in flower production would lead to an increase in plant size and or rubber yield. So by using what we know about the floral quartet model of flower development, we constructed several agrobacterium-mediated transcription factors that have the potential to repress the genes responsible for flower development, thus creating a potentially flowerless Wayuli. Next slide, please. Next slide. Thanks. The issue with transforming Wayuli is not the effective, oh no, go back a slide, please. Thank you. The issue with transforming Wayuli is not the effectiveness of agrobacterium infection, but in the recovery of potentially transformed explants. Since different varieties of our Parthenium species have variable successes with a single media protocol, careful attention to cali health is tantamount. Several factors weigh into the health of potentially transformed cali, such as temperature, light, nutrient concentration, time between media culture, and the techniques used to prepare and care for the explants. Thus, by using explants containing the leafy construct as a preliminary case study, we were able to identify areas of improvement in regards to light exposure and methods of tissue preparation. Next slide, please. In the preliminary light experiments, we tested groups of 6 plates, 1 being the top plate and 6 being the bottom plate on the left-hand side image. After every cali transfer from one media to the next, each potential cali was counted and recorded. This process took place within a period of 11 weeks where explants showing hyperhydricity were not removed. Afterwards, cali showing little signs of hyperhydricity were selected to advance further down the cali selection process. 96 plates were evaluated and show that at different time intervals, the cali under plates 5 and 6, or the bottommost plates away from the light, responded positively to lower light exposure and had a lower incidence of cali with hyperhydricity, where stage 7 is analysis at 11 weeks, now concerning the graphs on the right, and stage 8 is analysis at 13 weeks, once the false explants were removed. Considering the preliminary observations shown here, future light experiments will include quantifiable measurements of wavelengths and amplitude. Next slide, please. The browning of photosynthetic tissue after damage is a common occurrence in almost all plants and is primarily due to the effects of polyphenol oxidase distributed by the vascular bundles. Browning could lead to premature PPO, polyphenol oxidase, seeps into the media. In order to limit the amount of degradation, the optimized protocol instructs to make incisions around the vascular cambium mid-rib. This can coincide with the fact that that xylem and phloem do not contain nuclei apart from the phloem companion cells, where agrobacterium would show little to no activity. In addition, future preliminary experiments will be made to lower the concentration of PPO with a 0.1 milligram per mil amount of cyclohexamide medium. And for those of you who are asking questions, I'd ask your mic.
Mariano, you were losing your audio a little bit. You may want to try closing down the video. Sorry.
Okay. Is that better?
So far, but it is.
All right. Next slide, please, under achievements. Thank you. Thus far, we have succeeded in improving the protocols for cali regeneration, and putative leafy transformed explants were recovered from 3 independent events. Once the DNA insertion is confirmed against the leafy construct, the explants will be transferred into greenhouse conditions for phenotypic analysis to check whether or not they have flowers or the amount of flowers. In an effort to lay a solid foundation for future biotech work and genetic engineering floral dip transformation, plant transformation of diploid Wayuli, the development of a protocol for embryo rescue, identification and knockout of heat shock proteins in Wayuli, and CRISPR-Cas9 genome editing will be set. Plant transformation success in Parthenium has met with challenges in improving the efficiency rate and may take 1 year to fully characterize and confirm a positive transformant. Thus, a floral dip method for agrobacterium-mediated transformation for Parthenium species is being considered. This method can shorten the window between the analysis of positive transformants to approximately 3 to 4 months, considering the time between the treatment of agrobacterium to the flowering and seeding state to the red fluorescent protein analysis. Since this analysis requires the characterization of viable seed and seed viability is dependent on successful pollinations, bees will be employed for pollinations during a given period after the agrobacterium application. Germination of seedlings will be done and embryo rescue will be implemented if needed to recover potentially transformed seeds. Out of the future experiments, the floral dip transformation will take precedence, and developments in this regard will be applied throughout the rest of the experiments. Next slide, please. I'd like to thank and recognize the helpful publications from McMahon et al., our lab, Miss Catherine Asau, the best plant physiologist, Stonebloom and Scheller, Cornish, Ray et al., Dirick et al., Waynes et al., Dong et al., Twanjun et al., Blumel, Thyssen, Gore, and Ali Estelai. Information from these publications provided a clear scope of the developments that needed to take place in order to bring Waiuli into commercial production and gave clarity to some of the esoteric characteristics and physiological responses that our plant carries. This work shown here today is one of the many ways we use genetics to improve the biomass quantity and quality of Waiuli, while creating a foundation for future biotechnological applications and breeding for improved genetics and characteristics. Thanks, everyone.
Great, thank you. And there's a question in the chat from Katie Brewer, just 2 words. She says, explants, question mark, Cali, question mark.
Okay, so Cali is undifferentiated cells made from a section of leaf tissue that has been infected with the agrobacterium. So they are cells undifferentiated, and we use a couple of ways to increase either the shoot or the root cell development. Explants are the stage after cali. They are what looks like a plant, but they're not totally a plant. They're usually rootless, and we increase them and divide them from the same cali tissue that comes from the leaf. I hope that helps.
Great, thank you.
Okay, Mariano, thank you very much. Really appreciate that. Patrick.
Hi, Dennis. I'm here. Can you hear me?
Absolutely. Go for it.
Very cool. All right. Hey, everyone. My name's Patrick, as you said before, and I work with Kim Ogden at the University of Arizona. Today, I'm talking about modeling Waiuli. So adapting the aquacrop model that already exists and changing it for a perennial crop. Next slide. That's interesting. It shouldn't be blind. Oh, there you go. Okay. All right. So just before we start, the work that we do is pretty much right in the field. So with feedstock development, before it heads to transportation and eventual processing, all this work we're doing is taking in data that we collect via drone from Dia and Matt and the work that he does and collecting temperature, and we combine that in this model to try to predict what the biomass accumulation is going to be over time. All right, next. Okay, so going with that, why model, right? So there are lots of good reasons to model. First one being forecasting biomass and yield, like I said before, right? So that's important wherever you are in the feed chain. You can be a farmer, someone who's working with bioprocessing, or a manufacturer. Modeling allows you to be more flexible with changes in climate, or moving around your plants and harvest dates and crop rotations. It allows you to really optimize a lot of things, because sometimes you have variables that you can't change, right? You can't change the weather, you can't change the temperature in the region, right? But you can change the spacing between crops, your irrigation treatments, what type of soil to a certain extent, right? So these reasons all add up together and make a very compelling case for spending the time to model. All right, next. All right, so aquacrop. This is the model that we choose to use. It was developed by the Food and Agriculture Organization, United Nations. It's a water productivity driven model, right? So first and foremost, the most important parameter that it uses is water stress. And since we live in the desert and we're looking at arid crops, that was very important to start with. It's been successful in modeling, corn and cotton, and it's widely applicable. But one of the biggest drawbacks, it was not made for and intended to be used for perennial crops, right? So it was intended to be used for things that are easily swapped out year by year, to the end of the season. In addition to that, there's two versions. So there is a canned, I call it canned. It's a UI-based version that's an executable, right? It's easy to interact with, super easy to use, but it's non-modifiable. And then there's also a MATLAB version, which we'll talk about a little bit next. Next slide. Okay, so first of all, that canned version. Like you said, it's pre-compiled, easy to use, easy to teach someone pretty quickly how to use it. It doesn't take a whole lot of parameters. So compared to the other version, it requires, let's say 30 versus, it's much less customizable, but it makes it more easy to use and robust. It also has a GIS plugin, which is useful for, simulating different parts of your field at any given time. But as I said before, you can't really change it. Next. So the MATLAB version. It is an open source implementation of that canned aquacrop model. It was developed by Tim Foster, who is out of the University of Manchester. Like I said, fully open source, so fully customizable. You can change whatever you want since it's just MATLAB code. It's platform agnostic, right? So that means I can run it on my Mac, I can run it on my Windows machine, Linux, I can run it off a server, I can run it on my smartphone. So it's much more, accessible. But since it's more accessible, like I said before, it takes a lot more input parameters. In addition to that, the entire model itself is broken up into 54 separate modules. So these things govern everything from importing data to the model, processing it. There's, the different one for calculating canopy coverage, biomass, the amount of evapotranspiration. There's a total of 54 of these, and they can each be modified separately, which makes it really, easy to kind of keep track of what you change. It can also be run in parallel, which is nice for, trying to optimize things when you have to do thousands of simulations. And it's expandable. So instead of just modifying what you already have, you can add onto it, which is extremely powerful. And that's something that we're looking at doing. Next. So some example data that we put in the model include things like the soil properties, things that you can't really change a whole lot, like hydrology, soil coefficients, and thickness of layers and all that. And irrigation management. So in the model, you can specify percentages of flood and drip and how much of the surface is wet. Next. Next slide. There you go. Thank you. And then crop parameters, things that are really can and cannot be changed, right? So you can change when you plant, but you can't change the temperature that the Waiule starts undergoing dormancy, right? So you can take all these information and parameters and try to work with them, the ones that you can change to try to optimize what you're dealing with. Next. Okay, so the model itself outputs several things, but the most important of which are canopy coverage, water flux, in terms of how much water, something like the WINS model, although a little bit different, it gives you an estimate of how much water is actually going to be required by the plant. Biomass, root depth, and yields. So yield in this case, ultimately would be rubber. Next. So here's a general graphical overview of what the model looks like. So you see in the middle of the screen where all the arrows are kind of going towards, next to the number 1, it takes in the CC, which is the canopy coverage. ZR is the root depth, right? So it takes the canopy coverage and the root depth, in addition to weather data like CO2 and evapotranspiration, you see there on the top from the atmosphere. And it uses that to calculate that water productivity. So in the model, that water productivity is just saying how much biomass are you going to accumulate every single day based on how much stress you have in the entire crop. So starting from there, it goes to calculate that water productivity, which is used to calculate biomass, and that HI is that harvest coefficient, and that is it's pretty much a ratio between, well, it is a ratio between the yield and the biomass that you get. Next. So here's an example of, I don't have it labeled, but this is the Drip 100 irrigation treatment in Maricopa. So the black boxes are the model output, and the green curve, right, that's the aquacrop output. So this is for the CAN model. This is what we looked at in the very beginning. So as you can see, the green aquacrop model is monotonically increasing, but the data exhibits some inflection point between, July and August of 2018. So the model itself is saying that it's just steadily increase, but the real data itself shows it increasing sharply, dropping off, and increase again. And some of that is just because there's a lack of data between months 8 and 9 in 2018, as you can see there. But all of the irrigation treatments exhibit some of this type of behavior. So it's, the function inside aquacrop to begin with is just an exponential, right? So increasing exponentially up until it starts to decay, your typical S-shaped curve, logistic curve, but there's something a little bit differently happening there. So since we have MATLAB, we can modify it. Next slide. Okay, so this is for that same irrigation treatment at Maricopa. So what we decided to do is we were looking at it and say, hey, that looks kind of linear. So just to start with, we'll look at that. So this is showing the blue boxes and the connected curves. Those is the same canopy coverage data from the previous slide with the black boxes. But in this time, we added a best fit line for the first 200 days. And so we're saying, okay, that canopy coverage growth seems pretty linear. How can we change it? And so this is the idea. So I've also included a 95% confidence interval there, and we did these calculations for every single one, D50 to D150, including F100, and then looked at how well they fit. Next slide. So for the sake of brevity, here's a quick overview. So in the left column, those are all the irrigation treatments, right? And the first column from the left, that's the slope. So as you can see, starting from D50, D75, D100, the slope of that line, a best fit, slowly increases as you give the plant more water, which makes sense, right? So for the one we were looking at, that slope is around 0.6. It's pretty characteristic. So that's saying that the canopy coverage in that first period of time, the rapid expansion, grows at about 0.6% per day. So we can use this information and take that new equation and put it into the MATLAB model, which allows us to more accurately predict the canopy coverage. And since we have the canopy coverage, we can use that to, since that's what the model does, it takes the canopy coverage and uses that to calculate the biomass. So step one was really getting that accurate fit for the canopy coverage. Next slide. All right, so as I said before, immediately future goals, adding that linear fit into the model, and then fitting it to the biomass. That's really where we are right now. Which is going pretty smoothly. There's hiccups here and there, but it's not too bad. And then lastly is once you get a good fit, then you can start to play with the numbers, things that you can change. It's built into the model are a bunch of things that they call shape factors. And those really are a way of relating, two variables together. So maybe they're related via a straight line, maybe they're related via an exponential curve, something like a parabola, right? So you can play with that to see how the things change over time. Next. So lastly, I'd like to thank Matt for his temperature data in the field, Dia, of course, for all the stuff that he's gotten for me, last noted. And Bridgestone, in addition to them, Doug, Huntaker, and Dennis, too. Thank you for, showing me the way sometimes and providing some data here and there, so thank you. And that's it.
Thank you very much, Patrick. Are there questions? We actually have in about 5 minutes that we could do questions. We certainly want to give everyone time to get lunch, although most of you, the restaurant is about 10 steps from where you're sitting probably.
Yeah, exactly. There is a question in the chat from David. It says, will you validate by making a prediction for Eloy and seeing how the data corresponds?
Absolutely. So,
do you have a minimum temperature?
Yeah, so that's one thing we've been looking at recently is what the minimum temperature is. So this isn't part of this presentation, but somewhere else we plotted, the maximum, minimum, and average temperature in the field at Eloy versus date, right? And then we also look at the height of those plants over time. And what we can tell is that from before, our initial estimate was about 10 degrees Celsius, and that really hasn't changed. So in terms of modeling, they have the growing degree days is saying, if you're below a certain temperature threshold, that's the minimum threshold, that you will not have any biomass accumulation over that day. And so for now, we're sticking with 10C, but absolutely, looking at Eloy and then comparing that data to the actual canned model output. So remember, this is an open source version. So it's important to see that your results from the open source version match that of the original version. Because if it doesn't, there's an issue, right? So everything that we've done so far has, but we're kind of going into new territory, right? This adjustment of the equation is not something that you can do in the original version. So we're excited to see what happens now and definitely keep you updated.
Great. Thank you, Patrick.
You got it.
Okay. I want to thank Colleen for keeping us on track. Thank you very much for all your help. I want to thank Blase, Alonso, Hadiqa, Leah, Mariano, and Patrick for their work and their presentations.